When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
DNA, or deoxyribonucleic acid, right, the key thing to remember is that it is a polymer, right? And it's a polymer made up of these nucleotides, most importantly. So most DNA, as we know, is a double helix. This is where two individual polymer chains are lined up and then twisted around each other, right? So nucleic acids, right, or nucleotides, are make up these long chains of DNA and are joined together by phosphodiester bonds, quite importantly. So if we actually go and look at a nucleotide, it's made up of a few parts. So we've got this pentose sugar base, pentose meaning that it's got 5 carbons. Then it's got a phosphate group. Right, the phosphate group is important when it's bonding to other nucleotides. And finally, it's got a nitrogenous base. Right, so the nitrogenous base is either going to be guanine, cytosine, adenine, thymine, or if it comes to making messenger RNA, it would be uracil. So we actually go and look at the individual bases themselves that go and make nucleotides different from each other. So the key things are that the primary nucleic acids are adenine, cytosine, guanine, thymine, and uracil, like we've just said. So these are split up into purines and pyrimidines. So purine is kind of an aromatic heterocycle, right, because it's got a circular group composed of both carbon and nitrogen atoms. Right, purines include adenine and guanine. The other type is a pyrimidine. So pyrimidine is just a simple aromatic group composed of just 6 atoms, right, 2 nitrogen atoms and 4 carbon atoms. These are connected by alternating single and double bonds, like we can see on the diagrams. And this creates a relatively stable structure, right? So if anyone does chemistry, they are very, very similar to benzene rings. So we can see that our purine has 2 rings that are kind of stuck together, and our pyrimidine just has the 1 singular ring. So we just go and have a look at a quick question. So figure 21 shows a molecule of ADP, right? So draw a circle around the part of the ADP that is a purine. So if we were going to go and do that part, quite often when they go and they talk about nucleotides, they will go and relate it back to ATP or ADP. So if we were drawing a ring around the part that's purine, right, it's going to be the nitrogenous base pair. Right, the second part is to state 2 differences between a molecule of ADP and a molecule of DNA nucleotide that contains adenine. Right, the key important bit there is the adenine part, because effectively the adenine is what's going to make that nucleotide there. So there are a range of things that we can talk about. So there's some simple things, there's some things that are a bit more complex, but it's all about pointing out lots of those obvious things. So you can talk about the fact that ADP has 2 phosphates, whereas DNA has 1 phosphate. Remember, you can't just say one or the other, you've got to go make that comparison. ADP has a ribose, whereas DNA has deoxyribose. So we think about it, the name of DNA is deoxyribose nucleic acid. And ADP has a OH on carbon 2, right? So we can see there one OH there. And DNA has no OH on carbon 2 of its sugar molecule. So there's some of the key things that you would need to talk about.
So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.